Plenary for the day is Ivana Bozic, who is visiting us from the University of Washington. She'll tell us all about evolutionary dynamics of tumor progression.
Thank you. I'd like to start by first thanking Kristen and Sandy for organizing this wonderful conference. It's so good to be in person after so long. And for this opportunity to talk about our efforts to quantify tumor evolution. Before I start, I want to say that this talk is going to be very genetic centered. And that is not to say that other processes, such as what Andre talked about yesterday, for example, do not play an important role. But genetics is a key player in cancer, and it's easily measured and quantifiable. And that's what I'm going to be focused on today. So I think about cancer evolution, and I invite you to think about cancer evolution also as a stochastic process during which cells accumulate mutations that lead to tumor initiation, progression, invasion, metastasis, resistance to therapy, and so on. And these key players that are responsible for driving the cancer, we call them driver mutations, and they are depicted in this nice schematic here in yellow. And typically, we think that multiple driver mutations are needed for the appearance of an invasive solid cancer. Potentially, other driver mutation may appear later on after the first cancer cell has formed, and they can form subclones in this growing tumor. But in addition to driver mutation, that driver mutations, once you actually sequence the cancer, and as I'm sure you very well know, what you find is that the majority of mutations there are actually the so-called passenger mutations, which we think do not significantly affect the phenotype. Passenger mutations can be very useful as they can serve as, for example, a molecular clock, and we will be using them for that purpose as well. And I'll talk about that later on. But so what I try to do and what my group tries to do is to try to put some numbers and equations into this process. So what are the rates at which these mutations are accumulating? What are the timelines at which key events in cancer happen, and so on. And today I will be talking about two very different cancer settings. One is chronic lymphocytic leukemia, and the other is colorectal cancer. So I'll start with our work on CLL, which was a joint work with the labs of Cassie Wu from Dana-Farber and Getty Gatz from the Broad Institute, both in Boston. And before I talk about what we did, I'll give you a little bit of introduction, to CLL. So CLL is a B-cell malignancy, and it's a relatively common leukemia that happens typically in older adults. It is a peculiar type of cancer because in many patients, the treatment is withheld until the patients develop symptoms. And so watch and wait is the clinical approach once CLL is diagnosed. CLL is one example of a cancer that can be easily tracked because the white blood cell count does correspond well with the disease burden in patients. And so you see on the schematic that there could be this long period where the disease is slowly progressing, but once there's this kind of sharp increase tumor and increase in symptoms, a treatment might be started. But there is significant clinical heterogeneity, and in some patients, treatment needs to happen very soon after diagnosis. And we set out to understand why this is. Once treatment is started, it used to be combination chemoimmunotherapy, but nowadays it's many targeted inhibitors are also being used as frontline therapies. And you can see the effect of treatment very nicely on the patient's white blood cell count. It would drop sharply, but in some patients, after some time, CLL could come back, then it would be treated again, and so on. And so we set out to study the natural progression of CLL before this point of first treatment in untreated patients. And so it's a unique cancer where we can actually do that. And we had a longitudinal follow-up of patients, and we sequenced their CLL at multiple time points. But before we started even looking into the genetics and how it changes over many years, we noticed something peculiar looking just at the patient's white blood cell counts. And I was surprised to see that in many patients, the white blood cell counts, and you can see examples of two patients here, very closely follow very well-known equations of population dynamics, namely exponential and logistic growth. And so that was it was very satisfying to see real patient data follow so nicely these simple mathematical equations, where exponential growth can be characterized by just a simple fixed growth rate R. And logistic growth starts off with growing at some growth rate, but that we call also R, but then stabilizes as it reaches carrying capacity K. And so you can see on the left in patient 18, the carrying capacity was below 200, whereas in patient 21, it exponentially grew past 300. And so let me go into a little bit more detail into our patients. And this is the original discovery cohort that had 21 patients. And we are depicting their natural progression timeline on the right. You can see the time between diagnosis and first treatment, which was as short as 2 years in patient 12 and as long as 15 years in patient 18. All of these patients were treated at some point, and these filled circles correspond to times at which we were able to sequence the patient's CLL. We developed Bayesian methods to calculate the posterior probabilities of all parameters, including the carrying capacity, and classified based on that. And I'll talk a little bit more in detail about that later, but we classified each patient into these two categories, logistic and exponential. But we also noticed that there's like an intermediate category of patients that cannot be easily classified into one of these categories, maybe because their growth is aberrant or because we have insufficient follow-up. And so you can see that the logistically growing patients are depicted in blue, exponentially growing patients in red, and patients whose white blood cell counts were indeterminate are depicted here in pink. And one trend that you can, or that one can notice right away, although this is a limited data set of 21 patients, is that logistically growing patients tend to take longer to first treatment, whereas exponentially growing patients tend to take shorter. And this is something that we wanted to explore also in a larger data set. But before talking about that, let's dive a little bit more into the genetics, because the genetics is also interesting. And as we see, does tend to correlate with the growth patterns that we observe. So on the left, I'm showing all 21 patients, first logistic, then indeterminate, and so on. And also on the x-axis, you can see the known drivers of CLL. And you will see that for each patient, we have several of these rectangles. That's because we have several longitudinal time points where the cancers were sequenced. Also, the light gray means that the mutation was absent, whereas these colors from white to black correspond to mutations that are present in that patient's CLL, and the color corresponds to the cancer cell frequency. So black means that it was a clonal mutation present in 100% of cancer cells. And there's two interesting things that we can see from this analysis. The first, which we also show in a different way on the right, is that logistically growing CLLs tend to have, on average, a single driver mutation, whereas exponentially growing CLLs on average have 4 different driver mutations, which is a significant difference even in this small data set. So it is appealing to think that exponentially growing CLLs may represent a more aggressive form of disease that has collected more driver mutations. Another thing that may not be apparent right away, but as you look closer, you will see that there is not much change over the course of follow-up. So if we see a driver mutation in a patient at any time point, we will see it later and we see it before. So even though we followed some of these patients for more than 10 years, we don't see new drivers popping up suddenly. So it seems that the stage has been set for these CLLs some significant time ago in the past before they were detected. Okay, so those were the insights that we found from our original cohort of 21 patients, but we wanted to see if they would generalize if we look into a larger cohort. And for that, we looked to an already published data set of about 100 CLL patients that had sequencing at least at one time point. Because we saw that there is not that much change that is occurring over follow-up of a decade or so. And then we went back and looked for all of their white blood cell counts for these patients. And again, try to classify them into one of the 3 categories. And you can see on top, I'm showing several examples of logistic growth patients and exponential growth patients in this data set. And we were able to do exactly the same analysis in this larger data set. So for logistic growth above the little plot, we're giving the estimate of the carrying capacity, whereas for exponentially growing patients above the plot, we're giving the estimate of the growth rate as a percentage. And on the bottom, you see a little bit about what is under the hood and how we actually estimated and classified patients. So what we did was make a Bayesian framework based on a simple logistic equation. Because one can think of exponential growth as a special case of logistic growth with infinite carrying capacity. And we use that to classify patients into groups. So patients that had carrying capacity that was confidently below a certain threshold, which you see on the bottom left, were classified as logistic. And you can see the posterior estimates of the carrying capacity of logistic patients are very tight, meaning that really there is, the carrying capacity is felt in the white blood cell count data. Whereas on the right, patients whose carrying capacity was confidently above that limit were classified, and where basically carrying capacity was not felt at all, were classified into the exponential category. And we had this in-between category also of indeterminate patients. So the take-home message from this slide is that we were able to perform the same analysis on this validation cohort of 92 patients. So let's see what we were able to conclude. In this larger cohort, we could also find some and confirm some things that were only hints in our smaller data set. And those are that, for example, logistic growth pattern was associated with a particular genetic change, deletion 13q, which is known to correspond to more indolent CLLs, whereas trisomy 12 was associated with exponential growth and more aggressive CLL. When we looked at the number of drivers, we were able to again confirm what we found in the smaller cohort, and that is that logistically growing CLLs had significantly smaller numbers of drivers compared to exponentially growing CLLs. And finally, looking at need for treatment, because in this validation cohort, not all patients received treatment. So we looked at if we classify the patient into some growth category, how likely they are to receive treatment. And you can see that in the logistic growth category on the bottom, the large majority of patients did not need to undergo any treatment during our follow-up period, whereas exponentially growing patients, by and large, necessitated treatment. So this is all to say that kind of what we intuitively would expect, we do see in the data, and that is that logistically growing CLLs tend to be less aggressive, and that is seen here by the fact that they need treatment much less corresponding to exponentially growing CLLs. And there seems to be a genetic basis for this, and that is that logistically growing CLLs have a smaller number of drivers compared to exponentially growing ones. So before I conclude this initial discussion of this 2019 study, I also want to draw your attention to another interesting feature of this data set that we found, and that could potentially be a feature of CLL in general. And that is treatment-associated evolution, and how does that compare and correlate with the growth pattern that we found. Here we were somewhat limited because not all of our patients, of our original 21 patients, had sequencing follow-up after treatment, but some of them did. And so for some of them, we had data points where the CLL was sequenced after it relapsed after treatment. And so I give you here examples of two patients, patient 6 on the left and patient 10 on the right. Patient 6 is an example of an exponentially growing CLL. We had 3 longitudinal time points where the CLL was sequenced before treatment. Treatment was initiated around year 12 after diagnosis. And you can see this sharp drop in white blood cell count. But after about 2 years, the CLL comes back. And we had 1 time point where the relapsed CLL was sequenced. And what is interesting is that the colors that you see within this white blood cell count kind of cancer growing correspond to the subclones that are present in this cancer. And so you can see that the before treatment, the dominant subclone was this blue one. However, after treatment, the CLL that comes back is now dominated by the orange subclone, which was just a very, very small fraction of the cancer before treatment. So we see that there is treatment-associated evolution that happens in this exponentially growing CLL. And so by saying evolution, that doesn't mean that treatment caused it. It means that it basically lifted up one of the pre-existing subclones that was present at a very small fraction. On the other hand, for the logistically growing CLL on the right, we see that treatment happened after around year 4. And then we see that the relapse that happens around year 8, the CLL that comes back has exactly the same genetic composition as the CLL prior to treatment. So no evolution that happened here. And we were able to look into around 10, a little more than 10 patients where we had sequencing before and after treatment, and where we had white blood cell counts to classify them into one of the two categories. And we do see a trend of clonal evolution post-treatment in exponentially growing CLLs and no evolution post-treatment in logistically growing CLLs. And so this kind of makes me think that there's potentially something that is causing logistically growing CLLs to be more evolutionarily constrained. So that's an interesting avenue to potentially explore further. So this is a pretty rich data set where we have longitudinal follow-up of patients before treatment. And we wanted to explore it together with my former PhD student, Nathan Lee, who is now in Beipold in Berlin, to try to say something more about parameters that were driving these CLLs and the timelines at which key events occurred. And so to do that, we developed let me show it here. We started by developing a very simple two-type branching process of CLL or cancer evolution. And then we also expanded it to have multiple types and be more complicated. But in this, for simplicity in this presentation, I will be talking about just the simple case where we have a single parental population and within it, a subclone with a driver. And so this is a two-type branching process that starts from a single parental cell. And at some later time T1, a driver mutation occurs, founding the driver subclone, which is depicted in red. And the parameters of birth and death parameters are different for these two subpopulations. They are B and D for the parental and B1 and D1 for the subclone population. But what kind of makes even this simple two-type process already complicated is that we also allow passenger mutations to accrue. And for simplicity, we allow them to accrue at mutation rate U in both populations. And we assume that the patient is diagnosed sometime T later, and that tumor is bulk sequenced at 2 time points at or after diagnosis. And so this is kind of what we will need for our input in trying to understand the parameters of this process. And just to kind of bring home the point that there's a lot of heterogeneity in this process due to passenger mutations. You can see it depicted here where each yellow star represents a different passenger mutation. Blue is the parental and red is the subclonal driver population. And interestingly and nicely enough, we can show that in this setup, one can analytically estimate virtually all parameters of this branching process. So I will give you on the left the data that one needs to do it. This is, we need tumor sizes at 2 time points, driver cancer cell fractions, number of mutations in that first cell of the driver. And then we also need this important parameter gamma, which is the number of mutations that have frequency between, say, 1 and 20%. And this is because we want to estimate the neutral tail of mutations from which we can get the passenger mutation rate. If you have the data on the left, then we are able to estimate the following parameters on the right. Using relatively simple formulas, we can estimate the growth rates of the different subclones, the mutation rate for passengers, and basically all the times in the process. So time between first parental cell and driver, and then driver and a diagnosis. I will note that there is something that we are not able to estimate. So if you have ideas, that would be great. We can estimate the net growth rate, but we cannot individually estimate birth and death rates. So that's an open question still. And in order to see if our estimates are good and accurate, we ran a Gillespie simulation of the process and basically apply the estimates to see how good they are, because we know the ground truth. And here you can see a relatively typical example. We can estimate the growth rates very precisely. And we can estimate the mutation rate and the times accurately, but with larger variation. And I must say, this is not because there's something wrong with the estimates. This is because the stochasticity of the process itself. So number of mutations will fluctuate as they're kind of accumulating as a kind of a Poisson process. And so if we're dealing with a tumor that has a high mutation burden, then the estimates would be accurate. But CLL is a cancer that doesn't have that many mutations, so we do have this variability in estimates. And finally, after showing that the framework does make sense, we applied it to a subset of patients from the study that I talked about in the beginning. And why a subset? Well, first, our framework here assumes fixed birth and death rates for all cell types, which means that basically the subclones will be growing exponentially. And we know that some CLLs are growing logistically, so we did not want to apply the methodology to those. So we only apply it to a number of patients that are growing exponentially from the original data set. We were also limited by the fact that the number of mutations in some of the CLLs was very low. So we had to pick those that had a reasonable number of mutations to estimate the neutral tail. And as I said, our framework is applicable to not only a single subclone, but more subclones. It's just that the formulas become more complicated. And so here we see examples of two patients from the original discovery cohort, patients 6 and 9, that actually have the parental clone in blue and then 3 subclones each. And we were able to estimate the growth rate of all of them. But I thought the most interesting finding was our estimate of when actually the first parental CLL cell appeared. And in both patients, we see that this happened within basically the first decade of life. And this is what we see in other patients. So this is very interesting and reflects what was found in other cancer types, that it seems that the seed was planted very early. But I must say one limitation of this work is that we do assume fixed growth rates. So if there were significant changes in the growth rates, then one would need to take that also into account. But with that in mind, it's very interesting that it seems that the first CLL cell appeared very early in the patient's life, and they were diagnosed. The white line actually shows where the diagnosis happened, around age 50 and 60 in these two patients. And then the arrows show where the CLL was sequenced. Okay, so this concludes the first part of what I wanted to talk about. And I will now also give you some insight into a different study in a different cancer type. We wanted to understand the evolution of colorectal cancer, but now stopping at cancer initiation. So what happens before cancer initiation? And this was joint work with my former postdoc, Shay Patterson, who is now in Manchester, and Hans Clevers, who was at Hubrecht Institute at that time. And so the idea behind this project is to try to incorporate the biological knowledge that we have on colorectal cancer evolution. And let me remind you, and I'm sure you probably know, is that colorectal cancer is one of the most well-studied cancers because the samples are relatively easy to obtain. So what are some things we know about colorectal cancer? We know that there's some driver mutations that are significantly mutated in CRC. APC, the most well-known, is mutated in about 80% of CRC patients. And then TP53 and KRAS are present in about 50% of colorectal cancer patients each. And we also know this, there's this very well-known so-called Vogelgram of what we think happens during the evolution to colorectal cancer. And this process starts in the normal colonic epithelium that is characterized by this specific architecture that involves colorectal crypts. And then this tumor progression goes through multiple stages that are characterized by accumulation of these mutations, most important being APC, KRAS, TP53, and then increasingly changes in the crypt architecture, and finally ending in carcinoma that has completely lost normal tissue architecture. But we see that even in the adenomas, we do see kind of these remnants of the normal architecture. And so this is what kind of we think happens. The situation in reality is certainly much more complicated and more genes are involved. But this is kind of the schematic where of how we can think about cancer evolution in CRC. And so we wanted to have this biological insight as a building block of our model, where we wanted to have this multi-step accumulation of these 3 key genes as sufficient for the appearance of a malignant cancer. And so, so even though each of them is present in more than 50% of CRC patients, together they are present in about 15% of patients. So you can think of this model as being applicable to about a subset of 15% of CRC patients. And we also must take into account that APC and TP53 are tumor suppressors, which means that their normal function is to prevent cancer, and both copies of these genes have to be inactivated for them to become drivers. And then KRAS is an oncogene, which means that its normal function does not have to do with cancer, but if it is mutated at some particular positions, it can be activated and act as a driver of cancer. So in a sense, we need 5 steps. We need these 3 genes, which means we need 5 steps on the way to malignancy. 2 for each tumor suppressor and 1 for the oncogene. But what we also wanted to say is that wanted to include in the model is we did not want to assume any particular order in which these 5 mutations needed to occur. So we said, okay, what would happen if we just allow any order? Does that make any sense? Are we going to get something that is reasonable? And so you can see our model kind of depicted schematically on the right, where we start on the bottom left with a healthy cell that has healthy copies of the 3 genes. And then in any 5 steps you want, you can reach the top right, in which you have collected all 5 steps towards malignancy. And because there's multiple ways one can inactivate an oncogene, a tumor suppressor gene, the situation is even slightly more complicated than depicted here. So we actually have 50 different genotypes and 270 paths to malignancy to keep track in this model. So how do we parameterize it? we really tried hard to do it to parameterizing using data from patients or that are relevant to patients. So the first thing that we needed to determine were the mutation rates. What are the rates at which you would go from one node to the next? What is the rate of activation of KRAS or inactivation of a single copy of the APC gene? And we kind of used two key ideas. One is that we can actually count the number of positions where if a mutation happens, that could lead to an oncogene or a tumor suppressor gene. And here you see examples of two different genes from this Vogelstein review. On the top, PIK3CA is an oncogene. So you can see there's a small number of positions where all these red triangles, which means mutations found in cancer patients, occur. So you have to really mutate it at specific positions, and we counted those positions for the KRAS gene. Whereas RB1 is like APC or TP53 or tumor suppressor. And you kind of just need to make it not work properly. So you can basically mess it up by mutation anywhere as long as you're producing something like a stop codon. And so there's many positions in the gene where that can happen when we actually calculated them for the two genes in question. And once we had the numbers of driver positions in these genes, we also needed some kind of a point mutation rate to multiply it. And for that, we used this estimate from Hans Clevers's group in 2016, where they took healthy healthy stem cells for patients of different ages and expanded them into organoids and counted the mutations and kind of found that on average 40 mutations per year accrue in healthy stem cells of the colon in the entire genome. So we use that as our passenger mutation rate, or as our mutation rate, sorry, not passenger. And the final parameters that we need is, well, some of these mutations and some of these changes will lead to selective advantage. And selective advantage that we are most interested in is in the form of crypt fission. So for example, we were lucky that in the past decade or so, groups have measured the rates of crypt fission due to APC inactivation. And crypt fission is when a crypt divides into two daughter crypts due to APC inactivation and KRAS activation. And so we were able to use them as inputs into our model. And also we used as input the fact that TP53 was shown to not lead to selective advantage of its own in normal conditions. It was shown to lead to selective advantage in inflammation, for example, in colitis, but we are not talking about that now. And so this is all the input into the model. And so how do we actually model this? So we use two things. We use computer simulations, and we try to also calculate some things analytically. So in computer simulations, we start with 10 to the 8 healthy crypts and keep track of the numbers of crypts with all 50 genotypes, as in each run, there will be an evolution of these 10 to the 8 crypts happening. But we also keep track of the paths taken by individual crypts. So here you see 5 different paths that reach malignancy that are possible in our models. For example, first we could have a mutation in APC, then mutation in KRAS, then mutation in TP53, and so on. So many different options. But also what is important is that no crypt will, in most simulations, no crypt will reach malignancy within 80 years, which is what we looked at. So some crypts will only stop after 2 or 3 steps, for example. And so we also, so we keep track of this in the simulation and also analyze the system analytically. Okay, so this is our approaches to analytic approximations. And so these are semi-deterministic approximations that are valid in the limit of small mutation rates, basically. And we calculated, we wanted to look at 3 scenarios. Our first scenario is what if all drivers are neutral prior to all 3 of them being collected? Would completely neutral evolution be able to explain incidence rates that we see for colorectal cancer? Our scenario 2 is, what if only APC has selective advantage? What would happen then? And then scenario 3 is what we think is the reality, that both APC and KRAS have advantage. And then we have a formula for probability of malignancy at time t. And we wanted to compare these analytic approximations with computer simulations and also with some estimates of cancer prevalence, colorectal cancer prevalence in patients. Okay, so here we compare our analytic formulas, which are the solid lines, with computer simulations, which are these black pluses or X's. And we have a single data point, which is in red and represents the lifetime risk of colorectal cancer. So first, I would like to draw your attention to this bottom line, the blue line. This is our analytic prediction for fully neutral case. So if these mutations accumulated neutrally, prior to all 3 being collected and causing colorectal cancer, then our predicted prevalence at age 80 would be many, many orders of magnitude lower than the reported prevalence of colorectal cancer. So it seems unlikely that this is what happens. Even if we assume that just APC has selective advantage, being the strongest driver with the strongest selective advantage, this is still 2 orders of magnitude too low. So we really do see the need for both of these APC and KRAS to have selective advantage. And then we see that our simulation results, the top ones, do agree well with the colorectal cancer prevalence at age 80. I want to kind of point that this is just a single data point. And this is not to say that this explains everything in the entire incidence curve, but kind of we were happy that having our model being completely separate from incidence data with input from biological knowledge and measured parameters could be in the same ballpark and produce reasonable results that are comparable to prevalence of colorectal cancer. And when it comes to comparing simulations and analytic results, we see that they start to deviate for the full model at higher ages. And so now we have also worked out a more complicated and precise formula for the probability of malignancy that is fully stochastic in a recently published paper with 2 PhD students, Ruibo Zhang and Obina Okogu. So if you like math, you should check it out. We were also able to provide relatively simple estimates for the numbers of mutated crypts. And these are actually exact expectations. So this is the numbers of crypts that have a single driver mutation. And you can see that in the beginning, KRAS mutated crypts dominate. And this is because KRAS mutations only need a single hit. However, APC mutations take over around age 40. And this is because even though they need 2 hits, their growth advantage is higher than the growth advantage from KRAS. So they will take over after some time. And TP53 does not give any selective advantage on its own, and it's also needs 2 hits, so the number of TP53 mutated crypts is very low. We also calculated the numbers of doubly mutated crypts. And what is interesting here is that crypts that have two driver mutations, if one of the mutations is TP53, their numbers are super low throughout the lifetime. The only doubly mutated crypts that are expected to have to reach significant numbers are those that have both APC and KRAS mutated. And that is because both of these provide selective advantage. So this is kind of a hint as to how important selective advantage is to this model. And one final thing that I wanted to discuss that was interesting insight we found from this model is we wanted to see, well, we didn't assign any predetermined order to how these mutations should be collected. And so we wanted to see for crypts that reach malignancy within 80 years, what was the order of mutations that they acquired on that path to malignancy? And strikingly, we found that in 2/3 of the patients, we recover exactly the order that has been suggested by Vogelstein and others, where APC is the first to be inactivated, followed by KRAS, and then TP53 as the last mutation. So this was very interesting. And then the question was, well, why are we able to recover exactly this order? And my naive expectation would be, well, the order is determined by the selective advantages, meaning the fission rates of different provided by different mutations, and the mutation rates at which they are accumulating. However, what we found was that the order of mutations is completely, in our model, completely independent of mutation rates. And we could show this analytically, and we could show this also from simulations. So for example, on the bottom, we show the relative probability of different paths to malignancy. So we see there's multiple different paths that can happen. Rates 1 is our original rates, and then we multiply them by 2 or divide them by 2 or only multiply 1 by 10 to have it not be symmetric. And in all cases, up to stochastic fluctuations, we see exactly the same paths with more or less exactly the same probabilities. So this was super exciting to kind of find out that the order of mutations is determined solely, it seems, by the fitness advantages that they provide and not by their mutation rates. And I'm almost out of time. So to summarize, I hope I convinced you, and this is not an audience I need to convince, that mathematical modeling can provide insight into evolutionary and growth dynamics of human cancers in patients. For CLL, we found 2 growth modes, exponential and logistic, that are associated with different numbers of drivers, different need for treatment, and post-treatment evolution. And for colorectal cancer, our model that tries to recapitulate some of the underlying biology can recover the lifetime risk of colorectal cancer from epidemiological data. And as I said, the order of drivers seems to be influenced by selective advantage provided by them and not by their mutation rates. And this is my acknowledgement slides. So thank you for your attention.
I was wondering, with the colorectal initiation model, did you ever consider a model where heterozygous APC loss provides a fitness advantage? Could that ever be consistent with the data?
So we didn't because we did not think that it would provide fitness advantage, but it could be added for sure.
Cool. And have you ever thought of comparing it to sequencing of normal colorectal tissue? Because you can.
We would love to do that. If you can point us in the direction of where to look, that would be great.
Oh, sure.
Yeah, we'll talk later.
Thank you, Ivana. As I pointed out, I really like your CLL paper. I have studied it and I have already presented it in one of my lab meetings. I have I want to notice something about the indeterminate class. Here I saw that there are fewer time points in the data compared to the other two classes. And I was wondering this might lead to a misclassification-induced bias.
You're exactly right. Most of the indeterminate cases are due to insufficient number of data points. But we did not feel comfortable, confident classifying them into either of the logistic or exponential due to, fear of misclassifying. So that's why we have this indeterminate category.
And also another thing is that I noticed that there is a delayed exponential growth in some of these cases of the indeterminate case. And this may possibly suggest the presence of Valley effect. So you might consider of fitting a delayed growth model in such cases.
Exactly, yeah. So there's things like that. There's even more complicated growths that are happening. So yeah, certainly there's other effects that could influence that.
Hi, I have a question about the CLL and logistic and exponential growth. I am just wondering if you had a chance to try to introduce some of those driver mutations to, let's say, logistic or CLL with logistic growth and change the pattern, growth pattern, or vice versa, let's say.
So that would be like an experiment in mice, or we haven't tried it. I mean, our collaborators haven't tried it. We are looking more into using mice experiments, why CLLs may be exhibiting one or the other that is not just due to driver mutations, but it's still a work in progress.
Okay. Yeah, because I mean, it seems like the growth pattern could be used for chemoprevention, for example, studies just at early stage before even the treatment after the diagnosis. Growth pattern could be changed in patients by just simply giving some maybe targeted therapy later or earlier in life, so that it can have big impact on the patient survival, right? It could be.
Yeah, that's an interesting idea.
Maybe. All right.
Thank you.
Do you know if anyone looked at what's happening in the bone marrow? I'm talking about the CLL models. The interesting thing is the expansion is exponential, but it's taking over the course of the years. So I wonder, and yet sort of if you look at the stem cell cycling, and it's quite much faster. So I wonder what is happening sort of under the hood in the tissues that underlies this kind of relatively slow exponential expansion of the disease.
I would love to know. I think that's difficult to do in patients, but
bone marrow biopsy are doable, and I think
we didn't have them for these patients, but it would be amazing to look, because as you're completely right. The peripheral blood is not the whole story in CLL. It's just like easy to look at.
And also kind of a comment with, I guess, like you're already basically assuming there could be kind of infinite carrying capacity, but just like physical constraints must impose some carrying capacity on the leukemia expansion, because you cannot get more cells that could be fit into the body, and at some point I think it must be slowing down. Perhaps it's never reached because of toxicity or other issues, but like other kind of similar cohorts. So I wonder what is happening to these patients with leukemia growing exponentially. Are they dying of the disease before you can see any signs of the plateauing out? Or if you follow them up for long enough, there would be cases where it starts?
That's a good point. Obviously, there should be some carrying capacity, but the thing is that in these exponentially growing patients, that carrying capacity is unphysiological in a sense that they would be treated much before, like they would reach that. So until they reach super high white blood cell counts, we don't see any slowdown. So why is that happening? Yeah.
Thank you.
Thanks.
Thank you for a great talk. The data you have in CLL do you think that would be powered in terms of the number of samples where you've got growth rate data on all of them, or like carrying capacity on all of them? I guess the other thing, especially when they've got a carrying capacity, if someone's measuring that, just saying, oh, it's flat, but from your model fit, you've got the R. And then I guess in a lot of these patients, there's measurements on like depth of response when they were treated, or like duration until relapse. Do you know if you could see if there's any relationship between that growth rate and chemosensitivity?
That's a great question, but there are several caveats.
Yes.
First is, of course, that the CLL has been growing for a long time before diagnosis. And our model, if in logistically growing patients, is very good at estimating their carrying capacity, but it's not very precise when estimating their initial R.
Right.
So one would have to take those with a grain of salt. Also, we don't have follow-up for all of the patients. We do have for some. It's a super interesting thing to look at, but there's some caveats.
Yep. Thank you.
That was cool, Ivana. Obviously, I know you said the context you think is important, but I guess there has been a bit of discussion, especially in terms of the colorectal cancer work, about the clonal dynamics being impacted by the architecture of the crypt, like where the stem cells are, the shape of that. Obviously, you're not considering that. I don't know if you saw Ryan's presentation where he was talking about polyclonality versus clonality. What are your thoughts on that?
On the spatial?
Yeah.
It's a super relevant comment. And the architecture, I would love to understand the crypt architecture. And I would love to understand what exactly happens to it once you get, first mutation. How does that changes the crypt? What, as you get another one, how does that changes, how does messes up? But it's very like, I don't know. I would love to know.
Yeah.
But in order to put it inside a model, I would need to know more about it. I feel like I would need to put in a lot of assumptions that I'm not sure are well justified. And this is what's preventing me from going deeper into that. But I would love to understand the spatial effects.
So you think it's important, you're just not ready to commit to it yet?
Exactly.
Okay.